Welcome back, everyone. If you could take your seats, please. Thank you, everyone. So, for this next session, there'll be plenty of time for chat over lunch. So, for this next session, we're going to kick things off. So, this is the Abstract Award winners session. I think the sponsorship that we offer as UK Myeloma Society to attend international meetings, I think is a really important part of the remit of the UK Myeloma Society. We're obviously very grateful for our sponsors who have all sponsored the individual award winners, and I'll go through those in turn as those individuals present. So just to remind everyone that sponsorship is available for various meetings, so for ASH and International Myeloma Society and EHA, and we have regular calls for people to submit their abstracts. We're really strongly encouraging early career investigators to put in abstracts for us to review and to get sponsorship, particularly those who may be struggling to find funding to attend meetings. We know it can be very challenging to get funding, particularly for international meetings, so that's why we're very keen to support that. So please do take a look on the web pages on the UKMS website, which we'll go through that in some more detail. So we've got 4 award winners today, but all award winners for the IMS. We're going to kick off, first of all, with Rosie Taylor, who is presenting a paper regarding the role of multiple myeloma changes in fatty acid metabolism in tumour-associated macrophages and the role via a FATP2 dependent mechanism. Thank you, Rosie.
Hi, I'm Rosie, a medical student from University of East Anglia, and I'm going to be presenting my MRes research, which the generous travel bursary from UK Myeloma Society helped me present as a poster at EHA over the summer. So my project was multiple myeloma causes changes in fatty acid metabolism in tumour-associated macrophages via a FATP2 dependent mechanism. So first, a bit of background. This is just a few examples of some of the existing research which inspired the project. So there's more and more research in lots of different types of cancers looking at macrophages and how tumour-associated macrophages have a more M2-like polarization state, which helps support the tumour to fight it like the rest of the immune system, and how if macrophages have a more M1 phenotype, then that can help potentially fight the cancer. So for example, Yun et al. Found that in lung cancer, if macrophages had a more M1 type phenotype, then the tumour growth was reduced. And then one of the papers which has looked at in multiple myeloma found that a more M1 phenotype also led to increased cell death of myeloma cells. And looking at a key part of macrophage phenotypes is their metabolic pathways they use. And Su et al. Found that lipid accumulation metabolism and metabolism helped support tumour-associated macrophages in being tumour supportive. So for my project, I decided to focus on fatty acid uptake and metabolism, particularly focusing on extracellular transporters, CD36 and FATP2, and intracellular transporters, FABP3 and FABP5, which then allow fatty acids to go on and take part in fatty acid oxidation to help produce energy. So we hypothesized that myeloma causes fatty acid metabolism to be upregulated in tumor-associated macrophages. And the first step to address this was to look at gene changes in genes, like I mentioned previously, which are associated with fatty acid uptake and transport. So to do this, I isolated macrophages from the bone marrow of mice and then co-cultured them with mice myeloma cells, and did this both in direct co-culture and transwell system to look at the effects of direct cell-to-cell contact and also extracellular factors, then quantified the gene changes with PCR. So this found that FATP2, one of the extracellular transporters, when it was co-cultured myeloma, macrophage fatty acid transport through FATP2 the expression was increased, whereas CD36, another extracellular transporter, was significantly decreased. And then the FABP5 and FABP3, the intercellular transporters, both showed trends of upregulation. There was a significant difference in some of the genes in both co-culture and transwell, showing that direct cell-to-cell contact and extracellular factors both had a role in this. So after establishing the gene changes I found, I wanted to see if this was related to an actual increase in fatty acid uptake in the macrophages in response to myeloma. So to do this, I did a similar process with the same co-culture, but also added Bo-Dipi, which fluorescently dyes the fatty acids, and then quantified the fatty acids present with flow cytometry. So this found that while short-chain fatty acids were significantly downregulated, so there's less in the macrophages in response to myeloma, but the medium and long chain were significantly upregulated, suggesting overall there was an increase in fatty acids in the cells, in the macrophages, when co-cultured with myeloma. So after establishing there was more fatty acids actually in the macrophages, I next wanted to look to see if I could also find a change in metabolic activity in the macrophages associated with that. So as a similar co-culture, but to make sure I was just measuring the activity of the macrophages, I used 5-GM1, so the myeloma cell conditioned media, so I removed the cells before co-culture, and then did a seahorse analyzer to look at metabolic activity. So to measure metabolic activity, this analyzer measures oxygen consumption rate. So I found that at basal levels, there was no difference between the macrophages co-cultured with the conditioned media and not co-cultured with that media. But then when triggered, the maximal respiration those macrophages were capable of had significantly increased, showing there is an increase in metabolic activity. And I next wanted to try and establish what factors could be having a role in this mechanism. Based on the gene changes I found at the start, I thought FATP2 would be a potential factor in this, quite a good candidate to explore. So I did the same co-cultures, but added a condition with a FATP2 inhibitor, lipofumata, and then again, with flow cytometry, quantified the fatty acids in the macrophages. And this found, as before, comparing macrophages not co-cultured myeloma and co-cultured with myeloma, there was a significant increase in medium and long-chain fatty acids present in the macrophages, and then there was a significantly less amount of both medium and long-chain fatty acids in the macrophages if they'd been pre-treated with the FATP2 inhibitor and then co-cultured with the myeloma, showing that FATP2 does have a role in this mechanism. It didn't quite get down to control levels, suggesting that it's not just FATP2 having a role, but I wouldn't expect just one gene to be causing all these changes, so that wasn't unexpected. So in conclusion, I found that in response to myeloma, macrophages have an increased uptake in fatty acids and an increase in metabolic activity, and the increase in fatty acids was in part due to FATP2. So future directions I'd recommend from this is seeing if inhibiting FATP2 also has changes in macrophage function and metabolism, as I didn't manage to get to this during my year MRAs. And then also determining if fatty acid uptake also has a role in macrophage polarization function, and if these changes would also be present in vivo models as well. I'd like to thank Rushworth Group for helping me during my year doing the MRES, and again for UK Myeloma Society helping me fund going to EHA to present my work as a poster. Thank you for listening. Any questions?
Thank you very much. We'll just maybe a couple of minutes for questions. Just while you're thinking of that, I'm going to ask you to kind of extrapolate ahead based on that work, fantastic work. Do you see this sort of inhibition of these pathways potentially a role from a therapeutic perspective as well?
Yeah, so hopefully a lot further down the line this helps identify potential therapeutic factors that if we can see how this is supporting myeloma, we can then try to work out how to reverse it to help it attack, or like not support myeloma further down the line.
Great, thank you. Great.
Hi Rosie, very nice talk, thank you. A couple of questions to you. You used the 5TGM1 mouse, is a mouse line? Were your macrophages mouse?
Yeah, they were extracted from mice.
Did you try doing it in the human system?
No, I didn't manage to have time to do that, but I think that would be a good next step to see if that's also can be found in human cells as well.
And do you think the effect of the myeloma cells on FATP2 expression and fatty acid uptake is specific to macrophages, or do you think it's an effect that is seen in other cells, other non-tumor cells in the marrow microenvironment?
I think it's likely that could also be happening, particularly in other immune cells. I only looked at macrophages, but I could definitely see that affecting others with all the interactions in the microenvironment.
Thank you. Nice talk.
Sarah.
Did you look and see if the FATP2 inhibition had any effect on the tumour cells themselves, and the myeloma as well?
I didn't.
And their metabolism?
I didn't manage to look at that, but that would also be an interesting thing to look at as well.
Okay.
Thank you.
Great, thank you very much, and I should say thank you very much also to Menarini's Denmark who sponsored Rosie's award. Thank you. So the next bursary winner is Lucy Godsward from the University of Bristol, and her award was sponsored by Sanofi. And she's been evaluating the role of circulating proteins to multiple myeloma risk and looking at a Mendelian randomization study. So welcome.
Thank you very much. So yes, my name's Lucy and I'm a postdoc from the University of Bristol, and I'm going to talk about circulating proteins and multiple myeloma risk. So I'm going to have two parts of my talk. The first part, I'm going to give a brief overview of a Mendelian randomization project where we've estimated the causal effect of circulating proteins on multiple myeloma risk. And then secondly, I am going to talk a bit about a pilot study to validate the blood for biomarker discovery in multiple myeloma. So a bit of background. Multiple myeloma currently has no curative treatments, although treatments are always improving and there's some great options out there. And in order to come up with more curative, well, a curative treatment, more needs to be understood about the role of molecular traits in myeloma development. Proteins are specifically interesting to explore because they are the target of most drugs. And new technologies can measure thousands of proteins from small quantities of blood. Okay, I'm going to give a quick background about Mendelian randomization. So this figure demonstrates how it can be likened to a randomized controlled trial. So on the left, we have a conventional trial where you may have a population who are randomized to either receiving an intervention, such as a drug, or maybe in a control group. And in this example, if an intervention lowers levels of a protein or the function of a protein, and this reduces myeloma risk, then we can say that there is a causal relationship between that protein and myeloma. And this is because people are balanced by confounding factors such as BMI, smoking habits across those two groups. And in Mendelian randomization, we use a similar concept, but using the random allocation of alleles at conception. So if we have two genotypes in this example on the right, genotype B might lead to higher levels of protein than genotype A. And if people with genotype B have a higher risk of myeloma, then again, we can infer that there is a causal relationship there. And again, that's because people are balanced between the two genotypes by their confounders, such as smoking. And in order to do this, we need large genome-wide association studies of both our exposure, such as a risk factor, and an outcome, such as a disease. So the aim of my study was to estimate the causal relationship of circulating proteins on multiple myeloma risk. Our exposure in this analysis is protein levels, and the genome-wide association study data for this was firstly in UK Biobank, so 34,000 participants who had 3,000 proteins measured by a platform called Olink. And secondly, we used a population in Icelandic participants where they had over 4,000 proteins measured by a technology called SomaLogic, which is a slightly different way of measuring proteins. And in terms of the myeloma GWAS, the first one was in a Finnish cohort where there was over 1,000 myeloma cases. And secondly, using UK Biobank, which had over 500 myeloma cases. And in order to estimate the causal effect of the protein on myeloma, we take single nucleotide polymorphisms, so genetic variants, that strongly associate with levels of the protein to estimate the causal effect. And this genetic variant is not related to confounders, so it helps exclude confounding in that relationship. So on to our results. There were 440 proteins across both studies that had genetic variants which strongly associated with protein levels to generate 2 causal effect estimates. And this is shown in the forest plot, where I've highlighted 7 proteins which have evidence across a discovery and replication analysis for a causal influence on multiple myeloma risk. On the bottom axis, it's the log odds of multiple myeloma risk per standard deviation higher protein. And on the y-axis is the gene which encodes the protein. And I just want to highlight KDR, which is vascular endothelial growth factor receptor 2, where this positive point estimate suggests that higher levels of the protein increases the risk of myeloma. And in terms of what's known about these 7 proteins, the ones that I've highlighted in bold have had some implications in either the bone marrow or in cancer previously. Okay, and then I want to talk a bit about a pilot study. So I've got a little bit of funding to generate some data from samples from the Iceland Screens, Treats and Prevents Multiple Myeloma study to generate some inflammatory protein data using a company called Alamar. And my research questions with this, firstly, is how well does the blood plasma reflect the inflammatory environment of the bone marrow in MGUS? Because when we're doing biomarker research in blood, we're assuming that it accurately mirrors the bone marrow, and I want to basically assess the association because I'm not sure there's any evidence to show the relationship currently. And secondly, a pilot analysis to just see whether there are differences in inflammatory proteins across multiple myeloma, MGUS, and control plasma samples. And I'm hoping that this will lead on to potentially a bigger study looking at longitudinal changes in the proteome across MGUS and multiple myeloma. So to conclude, MR can be a really useful tool to explore the involvement of circulating proteins in myeloma. And our initial results from our MR study indicate 7 proteins which may be useful to explore further. MR does have its limitations, so generating more data from patient samples is really needed to further understand the role of circulating proteins in myeloma. And finally, I just wanted to acknowledge Matt Lee, who's performed some of these analyses with me. I'd also like to thank Myeloma Society and Sanofi for my bursary. I'm funded by Cancer Research UK in Bristol, and the picture on the bottom is me, my mum and me, and my mum is a patient with multiple myeloma, so I'd like to thank everybody in the room for your work.
Thank you, fantastic work. Got a question from Jonathan.
I might have got wrong in thinking about how these studies work, so from wrong. But you said it's similar to a randomised control trial and it's like an intervention, which is different. But can you be sure there's not sort of other genes which are co-inherited with that particular protein which can confound the risk later on?
Yeah, that is one of the limitations of MR, that there could be like linkage disequilibrium where a gene could be affecting another gene which might have other changes. So those are sort of some sensitivity analyses which we try to explore. But yeah, that's why multiple methods are important because each method has its limitations.
Charlotte?
Really interesting work, thank you.
I was wondering if your samples, when you measured
Sorry, I can't see. Oh.
when you measured the plasma proteins, were they taken from patients with myeloma or people who went on to get myeloma? I guess my question is, when you're measuring increase in VEGF-R2, is that just associated with myeloma as opposed to indicating a risk of it, which is what you implied?
So the genome-wide association studies were, the protein one wasn't performed in people with myeloma, so it's separate populations. So we're saying that in an average population, people with a certain variant have maybe higher protein, but in a different population, people who have that same variant might have higher risk. So that's another limitation, is that it's separate populations, so generating the same data in myeloma patients is important.
Okay, thanks.
And a follow-on from that is, do you think ethnicity will have a role to play? To use the Finnish group, that's obviously probably a relatively uniform population of individuals, making some assumptions it may not be, but do you think that it needs to be looked at in different populations to see the similar patterns?
Yeah, definitely. One of the limitations is that this was European ancestry, and the big genome-wide association studies just aren't really there in other ancestries, so that is definitely a focus where we need those big resources to be able to redo these analyses in other ancestries.
Thank you. There's another question, Sarah.
This might be the same question Charlotte was asking, so apologies, but if these blood samples were taken before people developed myeloma, do you think the protein associations were because the people already had MGUS, and that those are proteins that are higher in people with MGUS, or do you think they predict development of MGUS in myeloma? Do you think that difference matters in your question?
So I don't think we can really infer that from our MR analyses, but that's why generating the same or data in patients can help us disentangle that. But yeah, thanks.
I guess you need data sets
Yeah.
from the different stages.
Yes, one final question just over on the left.
Really nice, Lucy. I need to talk to you. We've also looked at the UK biobank data, but we've looked at it differently. We haven't gone down the route of using different alleles as you have. We've done a lot less sophisticated analysis, but we've compared markers that are present in the proteomic data in individuals that went on to get myeloma at least 12 months after entry to the UK biobank. And the longest one out that's gone out was, I think it was 9 years, and an average time point of about 5.5 years to get it. And we've pulled out a panel of protein markers that seem to associate with the risk of future myeloma, and they're a different set to yours. But there's very strong signal in ours are things like you've identified some of the proteins already have association with the disease. The markers we've pulled out, most of them are known to be elevated in myeloma. So to that last question, I think what we're picking up is an alternative signal for MGUS that's not immunoglobulin based. But it probably does signify the presence of an excess of plasma cells at a particular time.
Yeah, well, it would be interesting to see what your proteins are, because
Well, I'll show you.
the ones that we included had to have a genetic variant.
Yeah,
So a lot of them don't, so that could be why yours aren't appearing here as well.
I'd be interested if they're in the same pathway.
Yeah.
Great. Thank you very much.
Thank you.
Our next presenter is May Lowe. She is a senior pharmacist at UCLH. She's won the UKMS binding site bursary, and she's going to talk to us about barriers to the implementation of bispecific antibodies in the UK.
Thank you very much. Hi, I'm May. I'm a hematology pharmacist at UCLH. So I just want to, before I start my talk, I just want to thank the UKMS executive members, the UKMS and the binding site for sponsoring me to present our work on biospecifics at the International Myeloma Society meeting in Rio this year. So as we all know that prior to BCMA-directed T-cell engagers approval in NICE, patients can only access biospecifics through compassionate access scheme. or clinical trials, or self-funding. So, and also the dosing around bispecific and the close monitoring requirements during the initial phase does pose challenges for trust or for patients to access bispecific, and therefore a UK-wide multi-professional network has been formed to work on improving patient access, improving the equity to bispecific treatments. And the first task of our group is to understand what are the key challenges for UK hospitals to implement bispecific for myeloma patients. So we sent an online questionnaire through our professional network earlier this year to collect like qualitative and quantitative data. We do have 85 responses. For half of them were from tertiary centres, so this would be what you call the level 3 BSH care, and also level 2 and level 2A and 2B centres. We have 35 responses. However, we weren't sure whether we received any responses from level 1 care, because there were nine unknown areas of care centres that we received. So these are the main results. So we have a variety of healthcare professionals that have responded to us, and most of the centres, as you see, have access to chemotherapy, hotline service, A&E department, and also high dependent unit. I know this is a busy slide, so basically this is a collation of all the questions that we've asked people. So I suppose unsurprisingly, a lot of the level 2A and 2B community centres have limited experience in terms of treating patients with bispecific antibodies. However, they do have fairly good awareness of the trial data, the adverse events, and also the future use of bispecific antibodies in multiple myeloma patients. It is not sure, or we can't really tell, that whether the lack of e-prescribing support will be able to, is a key barrier to implementing bispecifics. We also look at different opinions from different healthcare professionals, also mainly to gauge their knowledge and also to help guide whether there is a education and training gap that's needed as well in other healthcare professionals. So from our survey, we found out that mainly the nursing team either will require further education, upskilling in terms of managing patients with bispecifics. We also asked centres to share their experience with us when they deliver bispecific through clinical trials, and I've done a thematic analysis on the positive and negative aspects of it. I just want to make sure that you know that this is done under the controlled environment and also very protocolized management of bispecific. So we have a lot of positive aspects around managing the complications with bispecifics. There are, unsurprisingly, capacity issues and also access to IVIG, for example, is one of the teams that's pulled through. So on that note, we also assess what are the key challenges around assessing immunoglobulin, and majority of our trusts had difficulty assessing it, mainly through commissioning restriction and also the complex approval process for accessing IVIG. We also asked for further comments on the areas of where the national group should be focusing our initiatives on, and the three main themes that actually shout out to us are going around the capacity issues in logistics and how to administer bispecifics, the need for clinical guideline or shared care policies, and also upskilling the workforce to be able to deliver bispecifics. So with that, our steering group has formed 4 main task groups. So the first one is on working on pathway redesigning. So that is the main focus on producing guidelines, care pathways, and IVIG subgroups with the main focus on working with commissioners to streamline the process of approval and also to review the current criteria for accessing IVIG. We're also working with the British Society of Immunology to produce best practice guidance on the use of IVIG in patients receiving bispecifics. There's also the third subgroup, working on patient and public involvement and engagement, mainly to understand the challenges in patients from minority group or underserved population, as where the challenges are with them receiving bispecifics. And the last subgroup is education and training, with the main focus at the moment on nursing or upskilling nursing workforce to help manage bispecific patients. So our pathway subgroup have recently published a consensus framework to help hospitals or centers to deliver bispecifics based on all the feedback that we've received through the questionnaire. So this is where you can find our document. And lastly, I want to thank the Embrace group members. As you can see, it's a multi-professional network from all over the country, and not to forget our patient and charity group as well. So thank you very much for listening, and I just want to thank UKMS and Binding Site for sponsoring me.
Thank you very much for the great talk. Any questions?
Sorry.
I was wondering if you had any feel from the survey as to what proportion of people were requiring immunoglobulin replacement?
No, we didn't look at that. We just mainly want to focus on the barriers and challenges from the providers in terms of delivering or implementing bispecifics. Yeah.
Thank you for that, May.
Hi, Alistair.
How are you doing?
Good.
I wonder if you're thinking about repeating that survey in a year or 18 months to see what the actual experience is.
Yes, we do have plans for that actually in next year. So we have a post follow-up evaluation, mainly to look at what the impact is of this framework if people have used it, and also what are the experiences in delivering bispecific since the NICE approval. We also have a pharmacy-led research, so led by pharmacists, principal investigators, to look at the access to bispecific or use of bispecific through a SEC data set. But that will be phase 2 of the project next year.
Can I ask a quick question? Just following on from the last question, do you think, if you repeat the survey in a year or two, do you think you will get, would it shift probably from delivery problems to things like IVIG delivery? Do you think that's going to be the main difficulty in the future?
I think with the framework we have tried to address ways to get around for capacity issues or resources. For example, how do you give ambulatory care? How do you prepare bispecific in the absence of a manufacturing unit? With regards to IVIG, we are working very closely with commissions at the moment to review the criteria in that hopefully we'll get a cohort approval akin like CAR-T so that it doesn't require such a strict criteria to access IG. So I can't tell for sure, but that's where we are working towards, and hopefully the best practice paper would help facilitate that as well.
Perfect. Thank you very much.
Thank you.
Thank you.
Thank you.
Our next talk is going to be by Chantel Hughes. She's a senior research nurse at UCLH, and she's the bursary winner of UKMS Johnson & Johnson. She's going to talk to us about participation in myeloma trials, whether this is representative of the UK population.
Hi, thank you for having me today. I'm going to talk about a data study I did looking at our chemotherapy data at UCLH. I have a habit of forgetting about my slides. I'm going to apologise at that up front. So as many of you in this room may know, myeloma disproportionately affects people from black ethnic backgrounds, with them being twice as likely to be diagnosed with myeloma than people from white ethnicities. Despite this, studies from the US show that our clinical trials data isn't reflective of this, with studies quoting around 4 to 6% of clinical trials patients being from black ethnicities, where in the US they would expect this to be closer to around 20%. And this includes the studies that we are using to register our drugs, so for example, pivotal CAR-T studies. To my knowledge, there's no large data set published of UK data, and given the differences in our healthcare system, we wanted to explore if this was a trend that we were seeing within the NHS. So to address this, I analysed our systemic anti-cancer treatment data for myeloma patients treated at UCLH over a 3.5-year period. I was looking at the demographic data of patients who were treated via standard of care and on clinical trials, looking at age, sex, ethnicity, and index of multiple deprivation, which we estimated from patients' postcodes. I did a multivariate logistic regression, which looked at sex and ethnicity. And this found that white patients were 2.5 times more likely to have been treated with a clinical trial compared to non-white patients. And this was a statistically significant result. We didn't see any difference for sex, but it is worth noting that there was quite a high proportion of missing data when it comes to ethnicity, with around a quarter of patients who were treated with standard care not recorded, and this was around 16% in the clinical trial data set. So quite significant gaps there. I couldn't include index of multiple deprivation or age in the multivariate analysis because there wasn't a sufficient sample size, but I did do an exploratory analysis just to see if there was any trends here. I didn't see any differences in the age of the two groups. However, there was a suggestion that perhaps trial patients are more affluent than those who are treated on standard of care. These findings align with the data we see in the US, which may suggest that the myeloma patients treated on our clinical trials are not necessarily reflective of the general population of patients. And that's even, in our system of the NHS. This lack of diversity raises concerns because it may impact the generalizability and the broader applicability of our trial results, and makes it more difficult for us to confidently inform patients of the risks and benefits of the treatments we're offering when they haven't been thoroughly tested in all of our populations of patients. It also risks contributing to increasing health disparities, which are unfortunately still on the rise in the UK. This analysis is obviously limited to a single site, but UCLH is one of the largest haematology units in Europe and based in the diverse city we're in today. So although it is a small data set, it may provide a valuable insight that perhaps this is an issue even within the NHS and the trials we run here. There isn't a lot of published research on why cancer patients are often underrepresented in clinical trials or what we should do about this. But the wider literature, some of the barriers that come up are things like mistrust of healthcare and research, cultural differences in attitudes to health, intersection with socioeconomic challenges and religion, And then things like inadvertent exclusions that are written into our protocol. So, for example, a recent publication highlighted how quite a lot of our trials that this publication had looked at have an inclusion requirement for a neutrophil level that actually would exclude people who have a Duffy null phenotype, which is more frequently seen in Black and Middle Eastern people. And these people would have a lower than normal neutrophil count, but they don't have any higher risk of infection risk, for example. It's also worth noting that our patient and participation involvement work is often also not representative of our patient populations. And, these are now required for most funding boards and ethics approvals. But it's hard to see how it is that helpful if we're not in designing inclusive research if we also don't have inclusive patient and participation involvement. Clearly it's a multifactorial issue and one that needs quite a lot of further research, but it's important in the delivery of equitable care for all of our patients. So thank you for listening, and thank you to UKMS and Janssen for supporting me to attend IMS to present this.
You've very eloquently identified what the reality is. How about overcoming some of those barriers? What would you say that the top thing can be done to try to improve efficacy in terms of recruitment to trials?
Yeah, it's tricky because I think, if you look at a lot of the literature on how we get to speak to and get involved with patients from, minority or underserved groups, a lot of the literature is about going into the communities and things like that, which is quite difficult with a rare disease. It's not like you can go down to the local community centre and you're going to definitely bump into somebody who's Black and has myeloma. I think maybe something that came up in some recent patient and participant work I did was about actually highlighting, does everyone even know what trials are? Do they know what we're talking about? And, certainly I think not that long history, there's some pretty unethical practices, and, that is within people's lifetime. So I think maybe, it's probably outside the scope of us here as haematology professionals, but I think general knowledge about what trials are and how we safely run them and how they can benefit people in their communities, I think might actually be quite helpful. Again, it's perhaps outside the scope of myeloma professionals, but I think it's something that's going to need quite broad approach if we're going to improve it.
So I would have three brief comments to make. One is your passion for what you're talking about is very clear. The other is that this is not a myeloma-specific problem, it's not a haematology-specific problem, it's a UK and probably wider problem. And I think one of the big drivers of the problem, as you look out in the room from where you are now, what does it look like?
Yeah, it's something that comes up again in the literature you read as places where they have a more representative workforce actually do see more representative recruitment. So I think actually coming back to your question, another thing I think we need to do is look at why are our research staff not representative and what can we perhaps do about that as well.
Thank you. Just a quick question. My name is Sally, I work in Bristol. Thank you so much for sharing the talk. Just to say really, I think it's extraordinary to do this analysis, such an important analysis on such a large cohort of patients. And if I may comment as well, from a personal perspective, the proportion of patients that your team are able to recruit into clinical trials is a significant number. But I just wonder if one of the ways of addressing this in the more longer term is rather than us doing trials as an option, it should be the standard of care for all our patients. patients. It's an opt-out in your utopian world in the future. So trials aren't the unusual thing to be entered into. They are the standard and there has to be a good reason not to be in a clinical trial. Now clearly, to reach that laudable, arguably unachievable goal would require an enormous amount of resource and a complete rethink of what we're doing. But it is, perhaps I'll speak a little bit to the Assurance report, which was published just last year, which really is a call to arms to really embed this practice within our trial networks. So thank you.
Thank you.
Thank you. Discussion there.
Sorry, I just run off.
There's one more question.
Can you tell I'm nervous?
So, thank you for a really interesting talk. And I suppose what we should think about doing now is thinking about the ways people access their knowledge and understanding of healthcare. So I think, one of the things that you can try to do is meet people where they seek their information. Now, I'm not going to be too flippant, but if we have more storylines in some of the TV programs that actually show someone who is of a black ethnicity, in, say, for example, EastEnders, which I have to give a disclaimer I don't watch, but if there are some of those storylines, tell people what actually happens, and to say, well, actually, there's someone here. We know that perhaps if a celebrity has breast cancer or prostate cancer, there's a lot more people who go for screening. And I think that's probably one of the ways we should use the modern technology or the world we live in now to get people to understand that this is an option. And I agree that making trials the default rather than the option would be another way forward. But yeah, let's sort of try and get people to understand this is part of normal life. Use storylines, use celebrities, and I think that would be really, really helpful. Thank you.
Thank you very much.